You can see my screen?
Yes, yes.
So,
Your
the topic
slides.
of
Your
discussion
slides.
today is, is how often you come across home mutations in genomic profiling, and why is it important to understand what is called as cis and trans. I will be going back and forth between few cases in the interest of the clinical community, why it is important to have cis and trans information. So one of the very classical examples where cis and trans is very important therapeutically is in the EGFR T790 position along
Dr.
with the
Vidya,
C797S.
Dr. Vidya, can you please share your slides again? We are not able to see. Just try again. Stop the sharing and then start again.
Can you see now?
Shh. Now we can see.
Yes. It full screen mode. So one of the classical examples where importance of home mutations and cis and trans in a given home mutation becomes an important factor in any clinical decision is the codon 790 T790M and the 797S in non-small cell lung cancer. Here essentially what becomes very important here is whether so if you see the distance between these two alphabets 790 and 797, there are 7 alphabets. 7 threes are 21, 7 alphabets in terms of amino acids. 7 threes are 21 nucleotides are apart. Now, if these two mutations happen in the same strand or the different strand, that is what essentially means by cis and trans. So, same strand is, as you know, when we do the sequencing of DNA, you keep adding nucleotides, right? So, first the T790M mutation appears and then the C797S on the same DNA segment. That is called cis. Suppose it did not appear in the same DNA segment, it appeared in another one where T790M is not there but C797S is there, then it is called trans. Essentially, it also infers that they belong to two different clones and not the same. So, here is one example. We know that we can look at this concept only when both the mutations are very closely spaced. If they are far away, means they are present in different exons, it becomes very difficult to understand the cis and trans relation for any two given mutations. So why is it important that this is all some fundamental knowledge? In the interest of time, I am not going to go into the details here. So basically, whenever there is a C797S along with the deletion, it is very important to see that they are in cis or trans. The reason why it helps when there is a trans is they can be re-challenged with first generation inhibitors along with third generation inhibitors. Unlike if they were in cis, they become much more resistant and they do not respond very well to any of the current existing lines of EGFR TKI as per the NCCN documentation. So essentially, why does that happen? Because C797S mediates the resistance to osimertinib binding pocket, basically. Because at T790M, it does not make any resistance to the osimertinib, whereas C797S develops a resistance to the binding pocket. Now, another, this is just an IGV to show how far they are actually. So this is the T790M location, T, okay. And after this G, this is where is our cysteine. Let's see the next slide here. Okay. So if both of them, I'll show you another graph where it shows up there. If both of them are in, if every, this is our DNA sequence in different colors. If both the mutations are present in the same line, that is cis. If they are present in two different lines, then it is trans. So here this is like this particular mutation, this is a noise, but essentially this mutation and this mutation are considered as cis because wherever this is present, this is also present. Now, to show it in a much better understanding, we have zoomed in to see these two locations, T790M and C797S. So here the middle alphabets get mutated, here the first or the second alphabet can get mutated to give rise to this one. So between these two, we have these many nucleotides, and this is where one has to pay a lot of attention to know how these clones are. This is not a mutant stretch which I am showing here, it is a wild type stretch. I'll show you few more cases where we have other types of co-mutations and how it becomes important. There is another important aspect that one has to pay attention to, that is the allelic burden. If you look at the allelic burden for T790M here, it's closer to deletion, and C797S is less, almost 1/3 of that. It is possible that not all clones that had T790M are carrying the C797S mutation. In other words, it can also mean that probably the cancer cell is going through a transformation and not all the cells have become completely resistant to osimertinib. That's another kind of an inference, indirect inference that we can gauge based on the allelic burden. Therefore, when you look at the cis and trans, you also have to pay attention to what kind of allelic burden is present to come to a consensus whether we have clones, all clones with these two mutations, or there are still some clones where only this is present and not this, or this is present and not this. So that is one more factor which one has to pay attention to in the clinical report. Why we cannot do this with any other exon, like 19 deletion cannot be having a correlation to T790M or C7 is because this is the exon 19, this is the exon 20, and between you have a long stretch of intron which we generally don't sequence. So this is your sequenced reads DNA on the NGS. You see that in between we don't read the intron because there is no information for us therapeutically relevant. Therefore, it is very difficult to correlate. Considering it is an FFP DNA, heavily fragmented DNA, we don't get to sequence long stretches of human tumor DNA. That's the reason it is very difficult to look for cis and trans, or are they, you can't infer whether the clone which is having the 19 deletion definitely carries all the clones carry T790M. That kind of an inference you cannot draw if the mutations are present very far apart from each other. Another classical example is between the exon 18, if you pay attention here, between around the 719 is an important activating mutation, but at times a rare one which is 709 can also happen. Here also it is exactly similar relation what we saw with the T7 and C797S. Here it is 10 amino acids apart, and therefore it means to say 16 nucleotides apart. Okay, 10 amino acids, 16 nucleotides is quite a big stretch. So it's very unlikely to see both of them together in a single strand. If the data quality is good and the DNA is not so fragmented, you will still get to see 16 nucleotides with abundant information on either side covered very well to say. And in other words, here again you can draw a conclusion. Mutant allele burden is very similar here. Possibility of both these mutations present in the same clone cannot be ruled out. Now, whether they are cis or trans is possible only if we are able to see them together in the same, in the same window where we want to examine the DNA, and the stretch should not be like fragments. Here you see the entire fragment goes from here to here, but what I am trying to show you here is essentially not as big as 16 nucleotides. So that becomes difficult to show within this window, and hence I have not put that example here. This is another example of exon 18 and exon 20 co-mutation. But here also, as I said, you cannot compare and come up with cis or trans because they present in two different exons. In between there is an intron. Nevertheless, the allelic burdens are very similar. Throws may or may not throw some light that possibility of being in the same clone cannot be ruled out. But here it is definitely not possible to give any conclusive answer on cis and trans. Unlike the other one where in the same exon 18 you had two clones, you can still look into it carefully. There may be few reads which will give you some insight whether they both were cis or trans. So essentially, why is it important to know? There is another important value add. When you try to kill the tumor cell with the TKI, that is a cytostatic inhibitor, not a cytotoxic inhibitor, if the abundance of both the clones are rapidly dropping together, means there is suppose a 1 log reduction in the liquid biopsy, you know that both of them are getting killed together in liquid biopsy when we look for an MRD. If one is getting killed faster than the other one, it's possible that they don't belong to the same. That's also one important application why one has to look and understand whether these two mutations are present together or away from, not from the same. So this is again an IGV example to show where these mutations exist. Again, they are far apart in two different exons, so it becomes difficult to document. So this is another example again, similar example what I showed in earlier case. Quickly going to, so this is the, as I told you earlier, how one has to look for it. This is another example where is a deletion plus these two, but here, okay, as I told you earlier, you cannot look for, cis or trans between the primary driver and the resistance because they are in two different exons, and here again you see allelic burden varies from primary driver to the resistant clones. That's the reason why I put up this case here.
Vidya, 1 minute.
Yes, done. So one important case here. Show the case then. There's an insertion. Yeah, here you see there is a driver mutation, but there is a VUS. Both of them are sitting very close to each other. With in the same exon 769 and 768, and both of them are in the same clone. You see, this is the mutation. Both are sitting close to each other, but you cannot infer that one would be also a driver. This is a VUS. This is only the driver. So that's also an important factor why you need to know whether it is cis or trans. I think I'm going to stop here. I have another very important case here. I just want to show that case. When you do real-time PCR, this becomes a very important aspect which NGS dominates over real-time PCR. Why one has to look at cis and trans and come up with a multinucleotide variant MNP as a answer. Before we go into giving the explanation here. So this is a case where we found at codon 13. If you see the variant color, it will give you a glycine to aspartate. It will also give you a glycine to cysteine. But we need to pay attention to the allelic burden. Both the allelic burdens are similar. Possibly that they both belong to the same clone. Let me look back. If they both belong to the same clone, that must be the same codon again. So if it were only single mutation, you will see single line. This is the single mutation change. Okay? But if it were both the mutations together, it is no longer what we interpreted aspartate and cysteine. It is totally different amino acid tyrosine. And it is very important to know how you combine this and look for the variant here, because unless you try to understand that these two are very closely placed variants and they both are always coming together, you cannot tell it. This is a cis configuration.
Yeah. Zoom.
Yes, done.
Yeah.
So, and the clinical relevance also will vary.
Thank you, Vidya.
Yes, ma'am.
So only one question if anybody wants, otherwise we'll...
Yeah.
So, Vidya, can you hear me?
Yes.
Vidya, I want to ask you, this cis and trans, of course, it is real time you will not be able to. You either need a have you seen like the response to like third generation and first generation, because the significance of this is main. So have you seen any cases of that sort?
Yeah, one case from northern part of India. I had a discussion again post-test, MTLB kind of a discussion with the treating physician, and the doctor also treated with re-challenged with first-line TKI, and there was a significant improvement. In that case, it was in France.
Okay. Thank you, Vidya.